All right, so it's good morning, everybody. It's I was going to say it's 9 o'clock, but depending on where you are, it's actually maybe a different time. So anyhow, so it's my great honor to introduce today's speaker, Valerie Chew from the Duke National University of Singapore Medical School in Singapore. She's a principal investigator in translational at the Translational Immunology Institute and an assistant professor at Singapore. Her research focuses primarily on understanding the complexity, diversity of the immune context of the tumor and microenvironment and its influence on clinical outcome. And I'm sure we will hear about this today and response to therapy in patients with HCC. For many years, she's been a pioneer in using novel techniques to conduct high and multidimensional immunophenotyping and immunomonitoring of the HCC microenvironment, using tools such as CyTOF, single RNA-seq, spatial transcriptomics. And this whole approach really led to a holistic profiling of immune landscape and the identification of clinically relevant immune substance and potential biomarkers predictive of tumor progression and response to therapy. She has done really outstanding work in the context of radiotherapy to HCC, and her work has gained recognition with multiple grants, high impact publications, and you go to any meeting and you will hear her either give a regular talk or just as recently the keynote lectures on the tumor microenvironment. And I'm super pleased that we have her talk at the at our seminar series today. And the title of her talk is going to be Targeting the Tumor Immune Microenvironment for Biomarker and Therapeutic Discovery in HCC.
Hi everyone, my name is Valerie Chew from Translational Immunology Institute, TII, SingHealth, Duke NUS, Singapore. So I hope you don't mind that I pre-recorded this video because just in case my home Wi-Fi can be a little bit unstable. Therefore, I thought it would be good that I use this pre-recorded video for my today's talk. And of course, I'm present and would love to take any of your questions at the end of my talk. So today I'm going to talk to you about targeting tumor immune microenvironment for biomarkers and therapeutic discovery in hepatocellular carcinoma. So just to begin, exactly what we mean by tumor microenvironment. And in fact, for the past many decades, cancer scientists in the field have been focusing a lot on the tumor cell itself, what we term the intrinsic factors. They try to figure out different mutation, tumor oncogene or tumor suppressor genes, and with that, using a targeted therapy or chemotherapy to treat cancer. But for all we know, that we have not found a cure for cancer, especially for hepatocellular carcinoma, which is known to be a highly heterogeneous cancer, genomically speaking. Therefore, we and others in the field are focusing more on the tumor microenvironment or the extrinsic factors. And our lab in particular are more interested in the immune infiltrates to the tumor microenvironment. Of course, we are also interested in the crosstalk between the immune infiltrates with the cancer cell. And with that understanding, we think this could lead to a groundbreaking therapy as well as the future of HCC therapeutics. Therefore, it's really time to talk about time. Time stands for tumor immune microenvironment. For today's talk, I will break it down to two parts. The first part, we will talk about our understanding of time in HCC. And as well as second parts, we will talk about biomarkers and therapeutic discovery. So first of all, we know that the HCC tumor is infiltrated with different immune subsets. And many of these immune subsets are either linked to poor prognosis or good prognosis. So for instance, the very famous regulatory T cell is very well known to be linked to poor prognosis. At the same time, some other cytotoxic T cells, such as the CD8 T cell, is often linked to good prognosis. And of course, we are talking about resection after resection. And in fact, we started looking in a cohort of Singaporean patients, 61 of them, in collaboration with Professor Han Chung To from National Cancer Center, Singapore, we found that a number of pro-inflammatory immunogenes are actually associated with superior prognosis in HCC after resection. So obviously we got the tumor sample from resected HCC tumor, and we look at the gene expression, particularly focusing on immunogenes, and we found those correlated to better survival are higher expression of a number of pro-inflammatory immunogenes, including TNF, and interleukin-6, CCL2, and so on and so forth. And we managed to validate these extended to 2 independent cohorts of patients from Hong Kong as well as Zurich in the Europe. So as we know that in Asia, particularly Singapore and Hong Kong, our patients are mainly hepatitis B viral-related HCC. But in the European patient, majority of the patients are non-viral in nature. And therefore, starting from our training cohort from Singapore cohort, we found these pro-inflammatory immunogenes related to better prognosis. And we are able to validate across another country in the HCC patient from Hong Kong and as well as Zurich, and showing that despite different etiology from the different regions of the world, these pro-inflammatory immunogenes are related to better survival profile in HCC patients. And more importantly, I would like to mention this paper. This is actually one of my best cited paper in my career. So we found that the interaction between tumor infiltrating B cell and T cell in the HCC tumor inside the tumor is actually related to better survival of the patient. It is able, we speculate, able to control the progression of HCC tumor. And if you can see that staining in red color CD20 and in green color CD3 T cell, either they form some kind of immune aggregates or more obviously like a structure or tertiary lymphoid structure. And we can see they're both related to good survival. Therefore, adaptive immune response in this tumor is associated to better prognosis in the patient. Particularly, this CD27 pro-stimulatory molecules is also a hallmark of T and B cell interaction. And it is expressed mainly on the CD3 positive T cell. And I would just like to mention this is one of the best immunohistochemistry images that I have throughout my entire career because it represents some kind of butterfly structure here. I thought this is just simply beautiful. And of course, CD27 being the co-stimulatory molecule, and we actually found them being very much expressed by the T cell that is interacting with CD20 and perhaps actually stimulating this correlation and the interaction between T and B cell. And this is together leads to a better survival of the patient. So as I already showed you in my earlier days of research, we show that active immune response or adaptive immune response in the tumor microenvironment is linked to better prognosis of the patient. And but of course, we also know that this will not go on forever. And most of the tumor sample that we analyzed before that are actually early phase HCC, early stages HCC. And we all know that with time and evolution, this tumor microenvironment will become immunoexhausted or immunosuppressive in nature. And you can imagine like a constant battlefield between tumor and immune microenvironment, and eventually the immune microenvironment will also become exhausted in nature. We also, through another study that's published earlier on, we found that intratumoral immune landscape are also heterogeneous when it is going through the evolution. It is a hallmark of evolution. So the immune landscape not only become more exhausted, it also become more heterogeneous, meaning to say one part of the tumor and the other part of the tumor, the immune landscape or the proportion become heterogeneous in nature. But of course, for the interest of time today, I'll focus to talk about more on the immune exhaustion and suppression in the HCC microenvironment, and how this can be driven by different factors, including hypoxia, chronic hepatitis B virus infection, and fatty liver disease. So first of all, I would like to introduce the immunoprofiling pipeline that we follow in the lab. So we not only look at the HCC tumor, we also study the adjacent non-tumor microenvironment, as well as the systemic blood from the same patient. We subject them to CyTOF. For those of you not familiar with CyTOF, it's actually a high-dimensional immunoprofiling technology, similar to flow cytometry, but instead of fluorochrome conjugated antibodies, we use heavy metal conjugated antibodies that can allow us to study up to 43 immune markers, both surface and intracellularly, on the single cell level. And as mentioned, I'm very interested in tissue architecture and the different immune cells residing in the tissue. We use multiplex immunofluorescence, or more recently, we venture also into spatial transcriptomic analysis. And as the whole of the tumor, we analyze using bulk RNA sequencing and also single-cell RNA sequencing. In collaboration with Genome Institute of Singapore, we also sometimes venture into analyzing the whole genome sequencing landscape, the DNA mutation. Together, we aim to provide a more comprehensive immune landscape analysis. So one of the earliest studies that we published using this comprehensive high-dimensional pipeline is we describe a more immunosuppressive environment in the tumor microenvironment, and it actually follows through a gradient where in the blood we see more naive type of immune cell, and in the non-tumor liver tissue we see memory type of cell that is not yet exhausted, but when in the tumor microenvironment we see an enrichment of exhausted immune subset. For example, the Treg cell that express more PD-1, as well as the CD8 memory T cell and CD8 resident memory CD8 T cell that express higher level of PD-1. And in fact, we found that not only PD-1, they express a number of checkpoint molecules, and which is including CTLA4, LAG3, and TIM3. This preferential enrichment or accumulation of CD8 T cell expressing checkpoint molecules actually point out to us that HCC tumor could be a good candidate to be treated with checkpoint inhibitors. And of course, true enough, we later on found out that through clinical trials, in fact, there's clinical benefits to checkpoint inhibitor for HCC patients. I would also like to mention this paper in Nature Cancer, where they found that both the resident and circulating memory CD8 T cell is important for durable response to immunotherapy. Pretty much coincide with what we found in HCC tumor, where both the resident and non-resident CD8 T cell express a high level of checkpoint molecules. Moving on, we then venture into study different factors that could drive immunosuppression in HCC tumor, and the first one being the hypoxia. So we know the hypoxia is a very harsh environment with lack of oxygen. Both the tumor cell and the immune microenvironment actually have to adapt to this harsh environment. So what we found from CyTOF is that there is accumulation of different immune suppressive subsets. And more importantly, I will just point your attention to two important types of immune subset. This is the regulatory T cell and CDC2 subset that downregulate HLA-DR molecules. So regulatory T cell, we know that is immunosuppressive in nature. However, CDC2 subset is well known to present antigen. But this CDC2 subset downregulate HLA-DR, which is the antigen presenting molecule, meaning to say they lose the ability to present tumor antigen. And we also found that this Treg and CDC2 subset that express low level of HLA-DR, they are very closely correlated with one another, particularly in the hypoxic microenvironment. To our CyTOF analysis, focusing on the doublet, which is cell and cell interacting doublets, we found that if we get them down, we know there are CDC2 subset interacting with CD3 positive T cell. And more importantly, when we further get down this T cell, we found that almost exclusively there are CD4 T cell, not CD8. And this CD4 T cell is actually, again, most exclusively Treg cell. We found this Treg and CDC2 subset doublet or interacting cell more significantly outregulated or enriched in the hypoxic microenvironment. Of course, seeing is believing. We stain for these two subsets in tumor microenvironment, and in the hypoxic tissue, we've marked by CA9, which is the hypoxic marker, and we found that these two subsets indeed are very close to one another. If we measure the distance between them, we found that particularly in hypoxic microenvironment, these two subsets are very close to one another with a very small distance between them, suggesting they are interacting with one another. So what do they do when they interact with one another? They actually undergo this phenomenon called trochocytosis, meaning to say that it transfers on one plasma membrane molecule to the other cell when they interact with one another. And true enough, when we subject this cell to in vitro hypoxic microenvironment, we see this CDC2 subset indeed lose the HLA-DR molecule. And they do so only with the presence of Treg cell. So as you can see, without the Treg cell, this CDC2 low, HLA-DR low CDC2 subset actually is significantly reduced. Therefore, the presence of Treg cell is very important. So where do this HLA-DR molecule goes to? In fact, we found that it actually got transferred onto the Treg subset. And we see the Treg subset actually upregulate HLA-DR molecule. And we also check the RNA expression level of HLA-DR on the Treg cell, and we do not see increase in RNA. Therefore, the only way that it gains this protein is actually to stealing away from CDC2 subset. In conclusion, we uncover interesting, unique immunosuppressive mechanism between the CDC2 subset and Treg cell under the hypoxic microenvironment. Of course, we are also interested in studying the different etiology and how they drive the immunosuppressive tumor microenvironment. First of all, we compare HBV-related versus non-viral-related HCC. Through our cyto data, we found that the immune landscape is rather distinct in nature. So for instance, we found that the HBV-related HCC are accumulated with resident memory CD8 T cell and Treg cell that express higher level of PD-1. And with that, we actually speculate that anti-PD-1 treatment will work better on HBV-related HCC because we already know that anti-PD-1 can reactivate the resident memory CD8 T cell that express PD-1. At the same time, in the same paper, we also describe anti-PD-1 could reduce the immunosuppressive function of the Treg cell. This will result into an overall enhanced anti-tumor immune response, particularly in the HBV-related HCC. And of course, this very controversial publication in 2021 in Nature indeed showed that viral HCC, particularly the HBV-related HCC, does seem to respond better to a number of checkpoint inhibitions. However, I would say controversial because subsequently, critical data analysis actually found not a lot of significant difference between non-viral and viral HCC, but I would still say that from what we found, at least from the T cell landscape, we know that it expresses high level PD-1, therefore the target for anti-PD-1 therapy. Of course, there are many other immune subsets that reside in the microenvironment. Therefore, I think there's a lot more to discover, and perhaps with the deeper analysis of spatial transcriptomics, we will start to see something even more interesting. Therefore, that actually initiated our next project looking into the NASH-related HCC. And so just to point out to you, the non-viral related HCC in this particular project are non-NASH in nature, majority of them, because these are Asian patients, and therefore we focus on steatotic liver disease-driven HCC tumor. And in this particular project, as I mentioned, there's a limitation on how much we can study using CyTOF and single-cell RNA-seq because we actually need to dissociate this tumor, and therefore the resulting cells are what we can actually dissociate from the tumor tissue. And to overcome this limitation, we actually use spatial transcriptomic analysis right now. So we have tried different technology, and currently we're still analyzing this data, and we hope to wrap up the story very soon. And for like a preliminary data, I can show you that we found that steatotic related HCC, and they downregulate the number of CD8 T cell, they accumulate more CD4 T cell, at the same time they accumulate more macrophages and have reduced level of B cell. And therefore suggests to us that adaptive immune response is actually reduced in the steatotic related HCC. At the same time, there are more macrophages and more Treg cell, and therefore we thought that they are more immunosuppressive in nature. As mentioned, we are working on the spatial transcriptomic data, and with that data, we now can actually look at rare subset that in the past we are not able to get because of the tumor dissociation problem. A lot of sticky cells such as fibroblasts are lost in the analysis. Therefore, we are not able to study them very well, but right now we can. And at the same time, we can study this cell-cell interaction because there are pieces of tumor we do not touch the cell at all. They're still in the vicinity, and we can study between one cell and the other how close they are to one another using several parameters such as the neighborhood enrichment score or cohort analysis, where we can study cell-cell interaction as well as the receptor and ligand pair that allow this cell-cell interaction. And of course, we are able to identify receptor and ligand pair that is specifically enriched in steatotic-related HCC. Again, this work is still in progress, and we hope to show you the complete manuscript very soon. Now I will switch gear to part 2 of my talk. I will talk about biomarkers and therapeutic discovery. So we were always hoping to get biomarkers to predict response to therapies for in HCC patient. And one of such project is actually targeting the radiotherapy in HCC patient. So this radiotherapy is called selective internal radiation therapy, or SIRT, and it is also called radioembolization. It is given as a single dose and is localized therapy injected directly to hepatic artery directly to the HCC tumor. It has a very short half-life of around 3 days, but whenever in the clinic, when we see a response in the patient, it is delayed, which is 3 to 6 month delay. And when they respond, they show a sustained response of more than a year. So this is in our patient cohort. And to an immunologist, it actually means an immunological response. And therefore, we collected the blood from the patient before and after Y90 therapy at a different time point. At the same time, we collect the tumor tissue when the tumor actually shrunk in response to Y90 therapy, and we get this tumor tissue from the responder patient, and we match them with a treatment-naive control tumor with all the matched clinical parameters. We analyze them using CyTOF and RNA sequencing. So first of all, when we look at our CyTOF data, comparing Y90 post-treated and responding HCC tumor, compared with the treatment-naive control, and we see the Y90 treated HCC that accumulated with a number of cytotoxic or killer type of cell, including natural killer cell, natural killer T cell, CD4, CD8 T cell that express activation markers. Whereas in the treatment-naive control, we see the usual accumulation of immunosuppressive subset. Majority of them are regulatory T cell. And that shows to us that this radiotherapy can actually enhance the immune response in the tumor microenvironment. How about in the blood? So because we are very interested to get peripheral biomarkers, therefore we also suggest the blood PBMC in CyTOF analysis. What we found is that, interestingly, we can see distinct T cell population in the responding patient. And these are actually a T cell with chemokine receptor, suggesting they have a tissue homing capability. And the chemokine receptor is actually CXCL6 and CCL5. They are present in the blood even before the therapy is given. So pre-therapy and up to 3 months after therapy, they are still very much enriched in the responding patient as compared to the non-responding patient. And because this is the certain chemokine receptor suggesting homing capability, we next look into the tumor microenvironment, comparing the Y90 treated and responding tumor versus treatment-naive control tumor. And we found that a number of immune-related pathway is upregulated in this tumor, and particularly the chemotaxis pathway in the Y90 treated responding patient. And lo and behold, we found the receptor that can actually bind to the T cell subset that is linked to the response to Y90 therapy. And these are the CXCR16 that binds to CXCR6, as well as CCL5 that binds to CCR5. And we find a very good correlation of this chemokine receptor, this chemokine expression with the T cell activation within the tumor microenvironment, suggesting that these chemokines indeed could actually attract this T cell homing into the tumor microenvironment and giving rise to clinical response. And apart from this T cell, we also observe this particular CD T cell that express checkpoint molecule PD-1 and TIM3, particularly enriched in the responding patients. And again, they appear in the blood even before the therapy is given and all the way up to 3 months after therapy. And with this, we actually initiated clinical trials combining Y90 with anti-PD-1 because we thought that this PD-1 is expressed in the responding patient and blocking them would enhance the immune response even further. Therefore, we collaborate with our clinical collaborators and run these clinical trials combining Y90 and anti-PD-1 in our patient with HCC. Of course, the rationale being multifold. First of all, we know that after radiotherapy, the T cell actually upregulate checkpoint molecules, including PD-1, justifying the combination with radiotherapy with checkpoint inhibitor. At the same time, we also know actually the tumor cell will also upregulate VEGF expression after radiotherapy. Therefore, combining with anti-angiogenesis, such as the adenosine therapy, will be also good as a combination therapy after radiotherapy. Now I've come to the last project that I want to talk about, which is identifying the biomarkers for response to checkpoint inhibitor and anti-PD-1 in HCC patients. For this particular project, we focus actually on two events. First of all, we know that objective response is around 20%. At the same time, the immune-related adverse effect is also around 20%. And we're talking about grade 3 to 4, which is a serious IRAE that can result into termination of the therapy. And therefore, we collected again the blood from the patient before and after therapy. In this particular project, we are lucky enough to also get tumor tissue before and after anti-PD-1 therapy. As mentioned, we are interested to look at the response and IRAE in the hope, with the aim that we want to actually understand the mechanism and design a combination therapy that could enhance the response without the increase of IRAE. And through our CyTOF data, again in the peripheral blood, we are able to find different immune subsets related to response and IRAE. Since we are interested in uncoupling these two effects of response and IRAE, we focus only on the immune subset that is common between these two events, which is the CXCR3 as positive CD8 effector memory T cell and the HLA-DR expressing antigen presenting cells. So first of all, we also would like to understand whether these cells do home to the tumor microenvironment. One of the first signs is that we do see this APC and the CXCR3 positive CD8 effector memory T cell reduced from the peripheral blood following the treatment trajectory, and therefore suggesting they could home to the tumor microenvironment. And indeed, when we look into the tumor microenvironment before and after checkpoint therapy, and this is only in the responding patient, we don't see it in the non-responding patient, upregulation of a number of HLA-related genes suggesting the homing of antigen-presenting cell. At the same time, they upregulate chemokines that could attract CXCR3 positive CD8 effector memory T cell. These are the CXCL9, 10, and 11 that binds to these CXCR3 molecules. Again, suggesting they are able to home this CD8 T cell into the tumor microenvironment. As a validation, we also refer to this Cell paper that published in 2021, where they studied more than 1,000 checkpoint molecule-treated cancer patients across 7 cancer types. And again, they found very similar signature. They found a CD8 signature, like what we found. And also they found CXCL9, which is again the chemokines that could attract the CXCR3 positive CD8 effector memory T cell, suggesting our findings is actually quite robust, even compared across different cancer types. In the HCC, we know that CD8 is indeed very important because in this other publication in Journal of Hepatology, we again find that CD8 plays a very important role in response to nivolumab, which is the anti-PD-1 treatment in HCC patients. So as I mentioned, we want to uncouple the effect of response versus IRAE. We know that we have CXCR3 positive CD8 T cell. This is a common immune subset that span across the response and IRAE. But we don't know actually exactly which type of antigen presenting cell that is related to response and IRAE because simply they are HLA-DR expressing myeloid cells. So through single cell IC, we are actually able to decipher this antigen presenting cell and found out that indeed it's actually a CDC2 subset that is related to response, whereas it is the CD14 myeloid subset that is related to IRAE. Therefore, that ends out that only one immune subset that is the true common immune subset between response and toxicity, which is the CXCR3 positive CD8 effector memory T cell. When we look at the different ligand receptor interaction signal in this particular subset, comparing response versus toxicity, we are able to identify the signature pathway that is only related to response or the one that is only related to toxicity. These are the TNF receptor 2 related to response and TNF receptor 1 that is related to toxicity. And to test that, we are able to target this pathway to uncouple the effect of response and toxicity. We treat them in the HCC mouse model. And so, as mentioned, TNF receptor 1 related to response to toxicity, and TNF receptor 2 related to response. And we combine them with anti-PD-1 or treat the mice individually with this receptor. So first thing first, we found that anti-PD-1 with anti-TNF receptor 2, in the mice, we observed absolutely no tumor at all. So showing to us that TNF receptor 2 that is related to response, indeed, when we combine this therapy with anti-PD-1, we see a much enhanced tumor control. How about TNF receptor 1 that is related to toxicity? So we look into the non-tumor liver tissue. We found that this particular molecule that is related to toxicity, once we block them, we see enhanced toxicity indeed. So we see enhanced CD8 T cell infiltration, and more importantly, this CD8 T cell express CD69, which is the activation marker for CD8 T cell. At the same time, TNF receptor 2 that is not related to toxicity at all, they are only related to response. And when we treat these mice, they have no tumor. At the same time, they don't show any sign of increased toxicity. So we can see that the CD8 T cell or CD69 positive CD8 T cell are not increased in the non-tumor liver tissue. With that publication, well, I would like to also draw attention to TNF receptor 2 because this particular molecule has been widely studied to play multiple roles in the tumor microenvironment. It can actually directly inhibit tumor growth as well as acting on Treg, MDSC as well as macrophages, and to reduce the tumor immunosuppressive functions. Together, this will make anti-TNF receptor 2 as an ideal combination for anti-PD-1 to enhance the immune response to within the tumor microenvironment. So as conclusion, I show you that pro-inflammatory microenvironment is good for prognosis in HCC tumor. And we also have shown you the immunosuppressive microenvironment can be driven by multiple factors, including hypoxia, chronic hepatitis B virus infection, and fatty liver disease. And I also show you peripheral biomarkers has got very high potential to predict response to Y90 radiotherapy to HCC tumor. And we also look into anti-PD-1, response to anti-PD-1, and also we propose a novel immunotherapy combination using anti-PD-1 with anti-TNF receptor 2 that will give us an enhanced response without the increase of toxicity. With that, I thank my lab members for contributing to the work, especially Samuel and JJ and Aldo, and my local collaborator, including Professor Pierre Chow, Professor Han Chung To, David Tai, and Shupin, and my local and other overseas collaborators. These are my funding information. I thank you for your attention and would love to take any question from you. Thank you.
Hi. Oh, sorry. Thank you. Finally, I was able to unmute myself. That was very, very interesting. So I have a general question for you, Valerie, which is, you have all this nice data, and you have upcoming, you show data on hypoxia, you have upcoming data on fatty liver disease, you have data on response to yttrium therapy. And then at the same time, you show this nice Cell paper where they actually looked at immune responses, pan-cancers. So what I'm missing somewhat is, is it always the same, or are there specific differences on, depending on whether this is in the liver or a specific treatment? Because I feel, in the end, we obviously, you need effector CD8 cells. You don't want to have too many regulatory T cells. You find increase in antigen presentation. Or is that a misconception?
Actually, okay, thank you for the very good question. So actually, I do feel that some of these signatures or T cell is rather important across different cancer types, at least for anti-PD-1 treatment. because I think the main target for anti-PD-1 is actually CD8 T cell. That's what I believe, and from our data and looking across other people's analysis, especially the Cell paper, they look across 7 different cancer types across more than 1,000 patients, and the most significant signature is CD8 T cell signature plus CXCL9, which is what we found related to respond to anti-PD-1 in the HCC patient. So I do believe this signature is probably common across different cancer types, at least for anti-PD-1. But of course, we are now in the era of combination immunotherapy. Then to me, that is a different story altogether. Right. So I guess we are still also looking at atezo-bev treatment and response. We are seeing a very different picture. So we don't see CD8 signature anymore. Instead, we see other signatures. So we have more macrophages coming in. We have CD4 signature, which have relevance to the resistance to the therapy and whatnot. So I feel that for combination immunotherapy is somewhat different, but anti-PD-1, I do feel that it is most likely a common signature that we are observing. Yeah.
Okay, are there anybody else that has any questions? Shen, yes.
Well, thank you, Valerie. I agree with Tim that really, so many data. It's a very exciting study. Thank you for sharing. A lot of data is, it would take me a while to dissect it, but I guess those are going to be online video recorded, and I can slowly go back again. So this is very good. I also have more kind of philosophical generic questions. So we know the tumor response to a treatment is mainly a tumor events driven. So now to a microenvironment, because the microenvironment is always there. It's a tumor that actually instigate the problem, and therefore there's some sort of interaction with the microenvironment. I'm just wondering what sort of a tumor features that you could actually identify. Now, the recent study that's showing the cell-free DNA is one of those, and they are very successful in using the public test. I think the trial called the Eclipse, something like that, and show quite strong features by taking the cell-free DNA, mostly those are from tumor, to model them. I just want to get your thoughts on what you think and whether you try those as well.
So I don't particularly work on cell-free DNA, so I can't really comment. And my main expertise or interest is mainly in the immune microenvironment. So I often just look at the immune cell. But in some of my study, I actually also look at DNA mutation, the different DNA mutational landscape. But again, I'm focusing on the immune response to this kind of mutation of the tumor cell. And I think with the era of immunotherapy, definitely the microenvironment plays a very important part. Whether or not different type of tumor or their genomic landscape will, is linked to the different response to immunotherapy, I think what is the strongest data out there would be like Wnt signaling pathway that's related to immune cold. Again, this is immune related. So exactly how much it is, linked to the microenvironment, in my own opinion, I think is quite important. So yeah, so because I don't really study tumor mutation, so I can't really comment on that. But I do think that microenvironment is extremely important.
Just to follow up on, do you know anyone actually is profile cell-free DNA for HCC for response?
I think there are a few publications out there. They look at this, and there are some preliminary data, I think, from a handful of patients, but not many. I myself don't do that, but I do know that in publication, I can't remember exactly which one, but there are people working on that for sure. I think probably Tim will know better, but yeah.
Thank you.
Thank you. Hi, there's a question.
Yes, Aitan.
Hi, thank you. Thank you for the wonderful talk. I just wanted to comment again on the general level. I don't know much about liver cancer. That I actually disagree. My hunch, we don't know, but my hunch is I actually disagree with you. I think there aren't very good general biomarkers, and it has to be carefully studied in the cancer type specific manner. For example, we have evidence that in some specific cancers, B cells and specific subtypes of B cells are more predictive of response to even to just to PD1, PD-L1 than CD8 cells, both in the blood and in the tumors. I also want to say that others and us have shown that actually there are some common predictors that are strongly predictive of which cancer types will respond to checkpoint therapy and which will not, starting from Liz Jaffe's 2017 New England Journal paper, and we have done work of that. But these biomarkers that predict which cancer types will respond are not at all predictive of patients' individual response within a cancer type. And when you think about it, it's a very different ball game. So I don't know. I think the jury is still out. I don't want to presume, but I think there are quite a lot of cancer type specific biomarkers that need to be studied. And of course, we need data, you know.
For sure, for sure. I guess I do agree in the sense that I think each individual cancer will have their unique and individual biomarkers that best predict response. So I'm just saying, you know, to my understanding of HCC at least, and to this other publication where they look at, you know, meta-analysis, it seems to have a common kind of signature that is more towards T cell, but like I say, I'm not saying that it will be the truth for all kinds of cancer. Probably each cancer will have their most predictive signature, which might not be T cell. But again, I guess one way or the other, anti-PD-1 is more targeting T cell in my opinion. So therefore, I guess if there's certain T cell signature that will predict response to anti-PD-1, that would make sense. And this is what we see in HCC. So yeah, of course, it may not be the, you know, truth for other cancer types.
I also want to say I was very interested and excited about your ability to distinguish the prediction of response and side effects, adverse effects. If that is true and could further be corroborated, that's extremely important because we want to treat earlier and earlier, so adverse effects prediction. But I think just my intuition that adverse effects prediction is much more complicated than response. And that is because the simple trivial observation that adverse effects occur across different tissues.
Yes.
And they may, essentially you need to build a tissue specific predictor of adverse effects.
In a way, so we look into the blood because we believe the adverse effect, the autoimmune disease could be captured in the blood. Because, you know, these T cells, they kind of circulate in the blood and it gives rise to multiple different tissue toxicity. And we include in our patient all the different sites of toxicity, but we take the blood T cell to analyze them. And what we found is that there's a commonality in the CD8 T cell that express CXCR3. And of course, then we chase after the tissue and find they are also there. But of course, we can't study all the tissues. So we only managed to look at the colon of the HCC patient who are treated with checkpoint inhibitor. And we do see toxicity in the colon, which is also one of the most common sites of toxicity. But obviously, you are right in saying that probably each tissue site of the toxicity might be different. But I guess, again, we are trying to find commonality and therefore our source is in the blood. But,
thank you. I'll make my last comment because I want, of course, to give others. So I just want to tell you, I totally agree with you that looking into the blood is where we should go for obvious clinical reasons and others. But I want to caution you that perhaps CD8 cells is not the best cell type to look into the blood. Because the turnover, we have a lot of evidence, but the turnover is not great because of the resident T cells and all that. So what you see in the blood is not necessarily what you get in the tissue. We have found that actually, again, B cells, to be or not to be, B cells B cells in the blood are much more predictive in the cancer types that we are studying. And others have recently reported that, so we are not the first or alone, and so on. So B cells have apparently a much better turnover. So you may want to look into them more carefully.
Of course, not discounting the impact of B cell, I can tell that you're definitely very interested in B cell. So I just want to maybe just make a point on the predictive biomarkers that we look at is pre-treatment. So they are already there, and what our hypothesis is that these are the T cells that home to the tissue, whether to the tumor or to the different tissue subtypes, where we then see the toxicity. So again, this is pre-treatment. So of course they are turnover, but that's somehow before the treatment, that's what we see, and they are related to response and toxicity. Yeah.
Thank you.
Thank you very much. If there's no further question.
I guess, thank you again, Valerie. That was amazing talk. Thanks so much, for presenting this late at night. We very much appreciate this.
Thank you.
And I wish everybody else here, a successful beginning of the week. Thanks.
Okay, have a great week. Thank you.
Great. Bye-bye.
Bye-bye.